Dear students, now I'm going to tell you about cell to cell interactions and cell surface markers. Cell to cell interactions, the ability of cells to communicate with one another. Communication between the cells. It's also called cell signaling that occurs in all multicellular organisms. So the cells in the body of a person, or else you can see the cells in any animal. So we will see they communicate each other through chemical signals. The cells of multicellular organisms use a varieties of molecules as signals. They may be peptides, proteins, amino acids, nucleotides, steroids, and other lipids. You have studied the protein hormones, steroid hormones. They are all chemical messengers. They synthesized somewhere else in the body or in some other cells, and they signal the other cells or target tissues to behave or to perform the specific function. So this is what you can see, the cell signaling. Water soluble signaling molecules cannot pass through this hydrophobic part of the membrane in the lipid layer. They bind to specific receptors, you will be able to see here on the membrane, and that is embedded in the plasma membrane. Cell must respond appropriately to extracellular stimuli to survive. So this type of the signaling is very essential for the survival of the cell and also to perform various activities of the cell. It depends on the extracellular signaling. So what are receptors? Receptors are nothing but the protein molecules. They may be present on the membrane, or else they may also present in the cytoplasm. They interact with these chemical messengers. So they may be hormones or neurotransmitters or chemical mediators, growth factors, or else cytokines of the immune system, etc. And they elicit cellular response. So these receptors, they may be present on the membrane, or else they may be present even in the cytoplasm. So they can bind specifically with the signal molecules and elicit cellular response. So cells coordinate their actions through different types of cellular communication. This is called cell signaling. Cell signals are chemical in nature. Hundreds of different chemical signals may be surrounding the cell. You may see here, so varieties of 100 and odd types of signaling molecules surrounding the cell. But cell responds only to signals to which cell surface receptor recognize. So the receptors present on the membrane can specifically recognize certain molecules. It cannot respond to the other molecules. It ignores the other molecules. So these signal molecules will bind only to those cells displaying the receptor proteins, which a shape into which they can fit. So it is just how exactly you have studied the the enzyme substrate interaction. Similar type, you can see the receptors molecules, they have a three-dimensional the structure where exactly the signaling molecule going to fit into this. So then it is elicit the intracellular signaling. So this binding induces a change in the receptor proteins shape, and that producing a response in the cell. So that is how it the from the extracellular the signal is communicated interior to the cell. So cell signal one another with chemicals. Receptor proteins embedded in the plasma membrane change shape when they bind specific signal molecules, triggering a chain of events within the cell. So this is a receptor. So you will find there is a chemical signal or a hormone. it is binding. So then it's a series of chemical signaling events going to take place, then it which ultimately elicit response. So cell signaling can occur between the adjacent cells, or chemical signals called hormones act over long distance. They may be they may interact with the adjusting neighboring cells, or else, for example, pituitary cells secrete some hormones which may interact, or which may have the which may elicit response in the target tissue, maybe a gonad or thyroid, like that, you have studied. So cellular communication is a multi-stage process that includes the following. First, the sending the chemical signal. So a particular cells produces this chemicals and synthesize these chemicals and get released into the blood or circulation. Then the target cell receive the signal, and you can see through the with the help of the membrane receptor. Then relaying this signal, the receptor relay this signal into the target cell's interior, either to the cytoplasm or to the nucleus, to elicit appropriate response. Then it brings some changes in the target cell behavior. So this you will find the you can see the hydrophilic signal molecule bind to the receptor present on the cell surface, whereas the hydrophobic signal molecules such as steroid hormones, estrogen, and you have studied such the glucocorticoids. So they may be directly, they may pass through this hydrophobic membrane, and they may have the receptor inside. They may have intracellular receptor, they may bind with that. So cells use different types of signaling depending on which other cells they want to reach. There are 4 types of cell communication. One is, you have studied synaptic communication. Sending and receiving cells have built a synaptic structure, bringing their cell membranes in close contact. It's called synapse. You know, the junction between the synaptic junction between the 2 neurons, or neuromuscular junctions. So where you can see to the exchange the signals easily through neurotransmitters, that is called synaptic communication. Then autocrine, this is what you will find here, the cartoon. It is similar. It is the signaling here. The whatever the chemical molecules synthesized by the cell itself, released outside, which again trigger, which going to bind on the receptor present on the membrane of that cell itself. So and it elicits the response. So this is called autocrine. Then the endocrine. Endocrine, the cells that is the producing the chemicals or hormones, they were somewhere else, and they release the hormone or the chemicals to the blood, and through the blood the target tissue will receive the surface receptor going to bind and elicit response. This is called endocrine. Then this is paracrine. So it is the signaling cell secretes a chemical that diffuses locally. It is the adjacent cell or neighboring cell. So it diffuses locally and reach the target cell. So this is called paracrine communication. Cells release chemical signals to regulate the activities of the other cells. So the actions such as cell division, cell growth, cell death, and the production of proteins. So all these are coordinated through the different types of cell signaling. So you will find the cell signaling may have elicit a survival response, or else growth response, or else mitosis or division, or else differentiation of the cell, or else apoptotic signal. So varieties of interactions, varieties of cell to cell interactions you will find. This is a synaptic communication. So the receptors are divided into four families. So they are ligand gated ion channels or ionotrophic. So these are the examples you will find here. So the ligand, when bind to the receptor, it induces ionic changes in the membrane. It changes in the membrane potential. So these are the voltage gated sodium and voltage gated calcium channels. You have studied last year how during the nerve impulse conduction and muscle contraction. Then G protein coupled receptor you will find. So it is there is a you can see the ligand here. The ligand means the molecule which activates the receptor. So it binds to a domain you will find here in this domain, and the you can see the receptor is coupled with the G protein, and it brings the protein phosphorylation, and which there will be a signaling cascade which reach the bring the intracellular effect. Then the enzyme linked receptor, that is the insulin receptor, growth factor receptor, cytokine receptor. So this is what you can see, enzyme linked receptor. So they bring the enzymatic action, you can see catalytic action. So once the ligand binds, it brings the catalytic action in the receptor molecule. Then you can see the intracellular receptors. So if the molecule or the ligand is a steroid in nature or lipid in nature, it can pass through this the hydrophobic region, and it binds in the intracellular receptors. So the group 1, 2, 3, these interact with the cell surface receptor. This one is the group 4, which can interact with the cells through the lipid bilayers of the cell membrane to interact these receptors found inside the cell. The receptor is present inside the cell. So the ligand passes through the hydrophobic region of the phospholipid bilayer. So this is about cell interaction. Now let us study about cell surface markers. So cell surface markers are proteins expressed on the surface of the cell that serves as a markers. You can see here the markers of specialized cell types. Cell surface markers are antigens actually on a membrane unique to a cell, serve as a monograms to help the identify. It is a how exactly all you people wear uniform of the college, and you will have individual identity card, thereby it is the identity of yours. Similarly, every cell or every tissue have their own cell surface markers or antigens, that is the identity card of that particular cell. So cell surface markers are special proteins and carbohydrates play a role in intercellular communication and recognition. So it is helpful in intercellular communications, how cytokines communicate, and also recognition you can see. So we can classify blood groups A, B and all depending on the cell surface markers that is present on the RBC. Cell surface markers are like a fingerprint specific to each kind of cell and capable of being identified according to what kinds of markers are present on the membrane. So it's like a fingerprint of a cell. So fingerprint of one person, it is it is not going to match with the other person. It is the identity of a person. So similar you will find. So this is the plasma membrane. You will find varieties of glycoproteins. You can see the cell surface markers you will be able to see. So these are different types of cell surface markers. They are vital for the body function. So the main class of cell surface markers are cluster of differentiation or as CD molecules. They refer to a system of naming of molecules of antigen. So they have developed a system of in naming these surface antigens, they are called cluster of differentiation CD proteins. So they are present on the surface of the cell membrane. The abbreviation CD followed by a series of numbers is used to denote the presence of the structure to which specific antibodies bind. So there are about 370 CD molecules in different cells. So they are named or numbered depending on the specific antibodies. CD molecules can act in numerous ways, acting as a receptors or ligands. They may function as a receptors, they may function as a ligands. The ligand is the molecule that activates the receptor. You have seen in the previous slide, which are important for the cell. Many cells express more than one CD marker. So many CD molecules are displayed on the surface of the cell. These are cell surface marker. You have studied immunology, where you will see T lymphocytes, T4 lymphocytes, where you can see the surface marker CD4 molecule. Then CD8 molecule, cytotoxic T lymphocytes. Just these are classified based on this marker proteins. The physiological function is being identified for many CDs. So this is very helpful in immunotyping. More than there are 370 CD clusters have been identified. Common cell surface markers are CD3, CD4, CD8, CD25. These are commonly used to identify the T cells. Their subcategories as a T cells ಅಲ್ಲಿ ಬೇರೆ ಬೇರೆ ಕೆಟಗರಿ ಇರುತ್ತಲ್ಲ, ಸೊ ಸಬ್ ಕೆಟಗರೀಸ್ ಸೈಟೋಟಾಕ್ಸಿಕ್ ಟಿ ಸೆಲ್ಸ್, ಹೆಲ್ಪರ್ ಟಿ ಸೆಲ್ಸ್, ರೆಗ್ಯುಲೇಟರಿ ಟಿ ಸೆಲ್ಸ್. ಸೊ to identify, so they are identified through these CD markers or CD proteins. So here you will see this is a cell, this is a CD molecule, and you can see it can bind specifically with the antibody. Antibody binds to a antigen or a cluster of differentiation present on the cell surface. So cell surface markers are special proteins and carbohydrates attached to the cell membranes. So they are special proteins allowing the transport of molecules across the membrane. They are of many category cell surface markers. They may be transport proteins, they may be antigens, they may be receptors, they may be ligands. They play a role in intercellular communication and recognition. Surface markers are like a fingerprint specific to each kind of cell and capable of being identified according to what kinds of markers are present on the membrane. So you will find this is the normal cell surface marker, and you will find the same cell when it is transformed into a cancer cell. You will find the surface marker changes, you can see. So they are displaying cancer associated antigen. This is how the prostate cancer can be. There is a prostate specific antigen will be displayed. Similar, you will find the cancer cells can be identified through their surface marker. Cell surface markers helps to identify the cell and distinguish from the surrounding population of the cell. So we can able to distinguish cancer cell from the surrounding population of cell based on these markers. These molecules serve not only as markers, but they also have a key functional roles. It is possible to diagnose the diseases. So it may be the cancer or the other diseases can be identified, and you can see the drug. It will helpful in treatment and drug discovery. Cell surface markers hold the potential aid to drug discovery. Cell surface markers has the potential to revolutionize the stem cell research and drug discovery. So this is what you can see, the stem cells can be identified depending on their cell surface markers, and how the markers will change as the stem cells differentiate, and all these are useful in stem cell research. So CD marker plays helps in the cell identification of the cell. It is like a cell phenotype. For example, B cell, they have all these CD molecules. That is the identification of the B cell. T helper cell, they have all these CD molecules, CD28, all these CD45, like that. Or else cytotoxic T cells, they have these CD markers. So this is how they are identifying identity of the cell, or else they are the cell phenotype, surface expression, you can see the CD molecules. So how we recognize people, they have curly hair, this girl is having straight hair, so this girl is having long hair. So similar, the phenotype, it's like that we can recognize the cell. The CD antigens are commonly used as cell markers in immunophenotyping. We can classify the cells based on the antibody binding. This is called immunophenotyping, allowing cells to be defined based on what molecules are present on their surface. Immunophenotyping, studying identifying surface cell markers will helpful to determine the internal structure of the cell. One can assess the what kind of cell it is. This is very important in disease diagnosis and drug discovery, and also it is very useful in research. So cell surface markers are many kinds of cells, and is related to the infection, different types of infection, and the development of several diseases also. So what are the functions of the CD? Just I not mentioned all the CD molecule, just for an example. So you will find that this is a CD, how exactly the surface markers, different types of molecules. So CD1, it is for the presentation of glycolipids to the natural killer cells, you know that. Then CD2, T cell adhesion molecule. CD3, signaling chain associated with the T cell receptor. CD34, hematopoietic stem cell marker. CD95, induction of apoptosis. So likewise you can see these CD molecules, they have a definite biological functions, and they are very helpful to identify the cell, and they are actually they are the phenotype of the cell. Thank you.